Welcome everyone. So here we again with another quick session on the topic genetically engineered insulin. So I hope, since this is a topic of class 12, I hope presently your board exams are going on. In fact, today was your chemistry exam. I hope it went on well, and I believe you still will perform better than your expectations in all the exams. And finally, the main aim is, definitely board exams are important, but our main aim is NEET. So for that, we are taking tests, we are having some doubt sessions, we are having the solutions, all those things are going on. But this is rather a quick revision session. I am trying to keep it short. This session speaks about genetically engineered insulin. So before we dig into the topic, genetically engineered insulin comes under the subtopic medical applications. In the topic medical applications of biotech, we have the first topic as per NCERT. We have genetically engineered insulin. The second topic we have is gene therapy, where we have already studied about severe combined immunodeficiency of that enzyme adenosine deaminase. And the third one is molecular diagnosis, where you have seen that ELISA, antigen antibody reaction that takes place. We check that in a spectrophotometer for color reaction. We have seen PCR. I have already said in the class the 3 steps of PCR are very important, be it board or be it entrance. The temperature, time, combinations. Now, our topic today would be mainly focusing on genetically engineered insulin, that too in a very concise manner. So, it is not that insulin is the only product that has been genetically engineered. See, insulin we all know it is used in the treatment of diabetes, specifically diabetes mellitus, and in that mellitus we see in diabetes we have administered insulin. So the problem underlying in the body could either be due to the deficiency of insulin or reduced insulin secretion from the cells of pancreas, beta cells of pancreas. There could be that the insulin secretion is normal, but the number of receptors are less or are non-functional, or it could be that body has developed some insulin resistance within itself. So based on that, we divide in diabetes mellitus as type 1 and type 2. In both the cases now, we see insulin is very beneficial, and diabetes is a very crucial disease, and the treatment is also, you can say, some customized treatment. I mean, we need to analyze the patient very carefully for the treatment. So rather than insulin, we have also produced, see here NCERT talks only about genetically engineered insulin. But if you want to know some extra points, we can say that other than that, genetically engineered factor 8 is also produced. Factor 8 is essential for blood clotting. We know the cascade pathway of blood clotting starting from factor number 13 to 1. So factor 8, if there is a patient of hemophilia A, you can administer him factor 8 because he has a deficiency of factor 8. Same, if he has a deficiency of hemophilia B, we can administer him factor 9, and these are all genetically engineered and produced. Human growth hormone. We have already produced human growth hormone by genetic engineering. Next, interferons, interleukins, all these substances have been genetically engineered today. If we see the data of NCERT, there are around 30 products worldwide that has been produced by genetic engineering, and out of these 30, 12 products are marketed in India. Is that clear? This is a point of your NCERT. There are 30 products that are produced, that has already been produced by genetic engineering technique, and out of which 12 is marketed in India. Now we are digging into the topic genetically engineered insulin. See, insulin we know is essential for treatment of diabetes. Now, if we see about the origin of treatment, originally insulin was extracted from source like cow, you can say from the beef. The other source was pig, that is pork. The insulin that was extracted from beef was known as bovine insulin. The insulin whose source was pork was known as porcine insulin. Actually, for pig we use the term porcine species. For cow we use the term bovine species. Cow, buffalo, we use the term bovine species. Bovine, the term means hollow horned organisms. The horn you see in a cow, inside it is hollow. So the term bovine is used for hollow horned organisms. Okay, let's get back to the topic. So originally the source was bovine insulin, that is from cow, or the porcine insulin, that was from pig. But there was a problem. One big problem was development of allergic reactions. This was one of the major problems. See, a patient having diabetes needs to be treated with insulin at regular interval. But if the source of insulin was bovine or porcine, although we have extracted insulin from their pancreas, but the problem was development of allergic reactions. And one more problem was that the efficacy, or you can say the effectiveness, was less. The effectiveness was not up to the mark. As per expectations, our expectation was that the sugar level should gradually fall in a certain proportion. When we use the term efficacy, means the expected recovery rate by using a drug is known as efficacy. If the drug, after getting administered in a patient, the patient recovers well, we said that the efficacy is fine. And if it takes some time or it doesn't function as per expected, we said that the efficacy is poor. So the efficacy or the effectiveness of the insulin whose source was bovine or porcine was not up to the mark, and the major problem was development of allergic reactions. Here we can also use the term hypersensitivity. So to reduce these things, we have to shift to a proper source. So this company, Lilly, this has itself produced, or rather we can call it Eli Lilly, this has already produced bovine insulin, porcine insulin earlier. But then they saw that these were the problems. Later they manufactured human insulin. Clear? So if the question says human insulin in 1983 was first marketed by, it is Eli Lilly, it is given in NCERT. But this bovine and porcine insulin, these were already present in the market at that time. The problem was there was development of allergy and the efficacy was less. Those were also marketed by Eli Lilly. So seeing the problems, scientists and researchers tried to find a solution. The solution was, say, this is a cell and this is the nucleus of that cell, and in this nucleus there is the DNA, and from this DNA scientists have extracted the insulin gene. Now before we see this process further, we need to know actually what happens in human body. In human body, insulin is usually in the form of pro-insulin initially or in immature form. Insulin is in the form of pro-insulin where it has 3 chains. It has 3 peptide chains, namely A, B and C. Now the term pro-insulin refers that they are inactive, or you can rather say it is going to mature. It is not matured, it is going to mature. These 3 chains are known as A, B and C chain. Say this is the A chain, first chain of insulin. From which arises the second chain, and say this is the third chain. So these are the 3 polypeptide chains. We call it this is the A chain, this one is the B chain, and this one, a connecting chain, is known as the C chain. And this C chain is present in pro-insulin only. Clear? When the insulin is not yet matured. From pro-insulin to matured insulin, this C chain is lost. See. Say this is the pro-insulin. On the process of maturation, the C chain is lost. What's remaining is the A chain. This is the A peptide chain, and the other one is the B chain. So in matured insulin, the 2 chains we see are A and B chain, and the C chain is absent. These 2 chains are linked by disulfide bridge. We already know what a disulfide bridge is. So these 2 chains is between these 2 chains, if the question asks the between the chains what bond we see a disulfide bridge. Also within the chain we also see disulfide bridges. Clear? Within a chain we find disulfide bridge. Between A and B chain we will also find disulfide bridges. Clear? So this is the structure of a matured insulin. So if the question says this is physiological process, this is not genetic engineering. Now what we are discussing, this is a physiological process. We will move to genetic engineering after a bit. So that I am recalling it again. There is an pro-insulin that is formed initially in the body. There are 3 chains, A, B and C chain. After maturation, when you see the matured insulin, the C chain is absent. Now we are telling that the chains are of peptide chains. So the peptide chain's length is how much? There is a definite length. There is a definite length. Okay, I am coming to the length a little bit later. So this is the basic process. I will discuss individual length of each of them, but a bit later. Let us see how genetically engineering going to produce this one. By the process of genetically engineering. See, this process takes place in the body. So by genetic engineering we need to produce this one. We will see what the process actually is. Say this is a human cell, and this is the nucleus. This is the DNA, and from that DNA we have separately isolated the A gene that will transcribe and translate to form the A chain. Similarly, we have isolated the B gene. Clear? Gene means actually what? It is a segment of DNA. Clear? Gene is actually a segment of DNA. So we have already isolated the A gene and the B gene. Now we will be introducing them into 2 different plasmids. Introduced into plasmid. We know what a plasmid is. We already studied about the basics of biotech, so we have an idea. So say this is a plasmid. This is the other plasmid. This is a plasmid where we will incorporate the A chain. This is a plasmid where we will incorporate the B chain. So we are taking that segment out and we are inserting our sequence. This is the sequence of A chain and this is the sequence of B chain. Now we will separately transform, or rather we can say introduce into separate E. coli. A very, very important term. The bacteria used is E. coli. So say this is an E. coli. This is another E. coli. Now within these two E. coli separately we are introducing this gene for, this is for A peptide, and say this one, this is for B peptide. Now this is genetic engineering. The process that we saw here, it takes place naturally in the body. It is a physiological process where the insulin is synthesized from a pro-insulin to a matured insulin, where the C chain is lost and the remaining A and B chain is connected by the, joined by the disulfide bridge. But this process, it is actually produced by genetic engineering technique. So now we have obtained A chain and B chain separately. Now in laboratory conditions, say this is A chain, this one is the B chain. So in laboratory conditions we form disulfide bridge between them, and this is what the insulin looks like when it is genetically engineered. The benefit of the process is that in our body during maturation there was no C chain, and when you have artificially or by genetical engineering, when you have produced this insulin, there is also no C peptide. Normally there are two chains, the chain A having A peptides and the B chain. So these also we have produced, that is the insulin now produced by genetic engineering is almost identical or similar to the insulin that is naturally present in the body, which reduces the chance of antibody actions against that hormone. See, when we introduce a foreign particle in the body, there is chance, or rather there will definitely be inflammatory response. But since although this is manufactured or produced by genetical engineering artificially, that is outside the body in laboratory conditions, but this insulin looks almost identical to the actual insulin that should have been present in the body. So it minimizes the risk of antibody action. Now when it comes to pro-insulin, there are three chains we know: A, B and the connecting C chain that is lost during the process of maturation. The length of A chain is around 21 amino acids, length of B chain is around 30 amino acids, where 30 peptides, and the length of C chain is around 33 peptides. But during the process of maturation, when we call it insulin, or rather we can say matured insulin as well, the C chain is lost, this remains the same. So if the question says the number of peptides in pro-insulin and the number of peptides in mature insulin, can we answer it? The number of peptides in mature insulin would be 30 plus 21, that is 51, and the number of peptides in pro-insulin would be 21 plus 30 plus 33, that would be around 30 plus 21 is 51, 61, 71, 81, 84. So by checking the length of the peptide, we can also say whether the insulin is a pro-insulin or a matured insulin. Clear? And but if it says there are 51 peptides, can you predict whether it is produced by genetic engineering or normally? No. When we want to check how much insulin the body actually produces, listen to this very carefully, this is a very conceptual term. When we want to check how much insulin is actually produced in the body for a patient who is already undergoing the treatment of artificial insulin, a diabetic patient, say his name is X, he is already been administered insulin, say for a few months. Now you want to check how much insulin his body produces. What would you do? See, the insulin that we are providing artificially, it contains A and B chain, that is the matured insulin. But the insulin produced in the body, it has been produced from ACB to AB. So we will check for the C peptide. We will check for the C peptide, number of C peptides present in the body. If the number of C peptides is less, that means the patient has natural insulin content very low. The insulin that is present in his body is because we are providing it in the form of insulin injections. But naturally, if we check the C level, if it is normal, his insulin level is normal in the blood. Because this C is synthesized in the body or in artificial insulin. We don't, although this is not necessary in matured form, but the matured insulin is produced from that pro-insulin. That pro-insulin has C1. So the C1 has also got some significance in the diagnostic purposes, but I don't think that would be very important from your examination point of view. But you can check the length of the peptide chains, these are very essential. Another question that is very important was main challenge in production of genetically engineered insulin. This is a line directly from the NCERT. See, when the question will ask you what is the main challenge for the production of genetically engineered insulin, the answer will definitely be something like the assembly. Assembly, what does that actually mean? When the matured chains are formed, A chain and B chain, they are formed separately in two different E. coli. This term is also very important, you have already seen in the diagram. Two different E. coli, one synthesizing the A peptide, other synthesizing the B peptide. So this part is okay, but the main challenge is to assemble them together to form the actual insulin structure. So this insulin was genetically engineered for the first time by a company called Eli Lilly, and I have already said this Eli Lilly already produced insulin before, that was the bovine and porcine insulin, but this time the source was from human, that was incorporated in bacteria. So that is why the name was given humulin. The insulin having the source of human, the name was given humulin, and you can say it is a revolution in the field of science. From human insulin, from human we have extracted insulin, we have made a culture, we have we have taken two different plasmids, we have taken two different chains, A gene and B gene, incorporated into different E. coli, assembled them in laboratory by developing the disulfide bridge, which was the main challenge. And this was made possible by the company Eli Lilly that already used to produce insulin from bovine and porcine source earlier. And then they gave the name humulin. It was in the year 1983 that they have got that success. This topic was very important because questions are very frequent from this topic. Even in the last NEET 2022, a very conceptual question was asked from this topic. In every alternate year, or in fact you can say in certain consecutive years too, we have seen the pattern that a question from insulin has been asked. Because see, the roots of this chapter also lies in chemical chemical coordination and integration from class 11, where you study about diabetes. So the point of explanation was that it is very essential topic and you must have a clear-cut idea. Let's check the questions. The first company to synthesize and market recombinant humulin in 1983 was, we already know it is Eli Lilly. Clear? Next, insulin chains produced by Eli Lilly using recombinant DNA technology actually taken from which of the following strains of bacteria E. coli? We have already seen that we took two different E. coli, one harboring the A chain and the other harboring the B chain, and then this A and B are joined by disulfide bridge. Next, the first human hormone produced by recombinant DNA technology was insulin. Which of the following peptide is present only in proinsulin and not in matured insulin? We have already seen in proinsulin we have A chain, then we have a C chain, and then we have a B chain. But in mature insulin we have A and B chain connected by disulfide bridge. So which one is absent? The answer would be C. The answer would be C peptide. Clear? Next, two polypeptides of human insulin are linked together by, we have already seen, this is a very easy question, disulfide bond. Humulin production through microbial factories does not include the incorporation of gene for humulin production. We take one A chain, we take one B chain, we connect them by disulfide bridge. We need A and B. The question asks does not include, which chain is not present here? C. The answer would be C chain. Clear? The main challenge for production of insulin was using rDNA technology was, as we have already seen in the last topic we have studied. The first is the getting insulin assembled in a mature form, it's correct. Getting insulin from human source, getting insulin synthesized chemically in proinsulin form, getting C peptide synthesis. The answer would be getting insulin assembled into the mature form. The mature form assemblage is tough. This part is also present in your NCERT book. In your NCERT it is given that, see here, the main challenge for the production of insulin using recombinant DNA technologies was getting insulin assembled into a mature form. Clear? See, if you see this diagram, it would be clear. This is the A chain, this is the C chain, this is the B chain. Once this is the proinsulin, once the matured insulin is formed, see the C peptide is freed out, the C peptide is lost. Now the A, this is the A chain, this is the B chain, they are joined by disulfide bridges. And I have also said between the chain will also be disulfide bridge. See here you can also see the disulfide bridge present. Clear? Okay, the question from NEET 2020, that was a good question. That question was good. Statement related to human insulin are given below. Which statement is or are correct about genetically engineered insulin? The question is not asking how many of the following statements are correct regarding insulin. See, the question is not asking how many of the statements are correct about insulin. The question is asking how many of the following statement is correct about genetically engineered insulin. First, pro-hormone insulin contains an extra stretch of C peptide. The statement is correct, but this is not relevant to genetical engineering. This process takes place inside the body. Are you getting my point? This statement is correct. This is a physiological process that takes place inside the body. But the question asked which statement is correct about genetically engineered insulin. That's why it would be wrong. Clear? The statement itself is correct physiologically. Second, A peptide and B peptide chains of insulin are produced separately in E. coli, correct, extracted and combined. This one is correct. This one is correct. Insulin used for treating diabetes was extracted from cattle and pigs. This is not an application of genetically engineering. This is not regarding the human insulin. Is it clear? The statement is correct about the origin source. One was cow, one was pig, bovine and porcine, but not in context to human insulin that is produced by genetic engineering. Clear? Next, pro-hormone insulin needs to be processed for converting into mature and functional hormone. This statement is again correct, but again we don't mark it because pro-hormone, that is pro-insulin, is present in the body, not in genetically engineered form. Some patients develop an allergic reaction to foreign insulin. That is also correct, but not relevant to genetic engineering. The question was very controversial after the paper got done. Different students had different opinion, but when the answer key was given, the answer key said that only B is correct. This question is from set, but I saw it in question number, the paper set was S5 and the question number was 196. In that question, the problem is all these statements are correct regarding insulin. Regarding insulin, all these statements are correct, but the question said genetically engineered human insulin. That is why only B was given the correct choice. So the answer would be B only. So this type of tricky questions they ask not on your knowledge, but they try to make the language a bit difficult. You need to be very aware of this. Clear? I hope your exams go well. Every one of you perform well, and if it would have helped you anyway, I will try to provide some more sessions, quick sessions. Although this wasn't very quick, as I can see, it's been over 30 minutes. So I tried to conclude it within 20-25, but it took over 30 minutes now. So hope this helps. I wish you all the very best for your board exams, and prepare well during the board and after the boards for your NEET, and perform. Try to take all the tests very seriously, because the game is up to May. You have to perform well on the 7th. Clear? Okay, thank you. I hope this helps.